Singh. I am working as an assistant professor in HR group of institutions Ghaziabad in pharmacy department. Today I am going to teach you industrial pharmacy second with code BP702T. Today's topic is drug development and non-clinical studies. So let's see. Drug development. As you know, what is drug development? To develop drug during various stages. And what is teams? So it is a process of bringing a new pharmaceutical drug to the market after a lead compound has been identified by the process of drug discovery. It means when we select a drug, we have to make any drug. After drug discovery, we make various steps for development of drug, and our final aim is to enter the pharmaceutical drug into the market. For this, various stages involved in the drug development are: number 1, drug discovery and target validation. First of all, we do drug discovery and its target. Next is preclinical testing. Preclinical testing before clinic. Next is investigational new drug application filing. Next, clinical studies, which includes phase 1, phase 2 and phase 3. New drug approval application filing. This is also known as INDA, as you know, and this is known as NDA. Next is FDA review and decision. FDA is also known as, it is the US regulatory authority known as Food and Drug Administration. And phase 4, clinical studies. Next is, and because of different phases involved, as we have seen earlier in earlier slide, we need a team. That's why it is called as drug development team. So the team do discovery and development group, non-clinical group and clinical group. Clinical group do clinical trials on humans, non-clinical group do clinical trials on animals or in labs. Next is regulatory groups. Next is marketing group. Regulatory group also regulate drug when they are after post approval surveillance into market. Next is engineering group, which handles machines. Next is legal group, which make legal documents for approval of drug, and various other teams. Next is on which factors the success of any drug development team depends. So the success depends upon plan and replan carefully. Whenever or when you want to start any work, first of all you make a plan or replan it very carefully. Next is execute the plan appropriately. Next, hire the people who want to get the project done. It means make sure you are hiring the people who are desirous or who take interest in developing the drug. Next, differentiate what is necessary and what is not. Means various experts from various groups like regulatory groups, engineering groups, clinical groups, non-clinical groups, the experts of different areas, they make sure what is necessary for the drug development and what is factors are non-essential. And last and very important is have patience and have faith. Next is non-clinical drug development. What do you mean by non-clinical drug development? It means it is a stage of research that happens before clinical studies, also known as preclinical studies. And first of all, what is clinical studies? Clinical studies mainly when we do clinical trials on humans, this is called clinical studies. And what is preclinical studies? The studies we do before conducting clinical trials on humans is known as preclinical studies or non-clinical drug development. What is the objective of non-clinical drug development? Why don't we do a directly clinical trial on humans? So, because the main objective is to confirm that either the target drug is safe for use in human or not, done by studying animal pharmacology and toxicology testing. In non-clinical studies or preclinical studies, we do to check either the target drug is safe or in humans, or by doing animal studies. It means the preclinical studies done on animals. We do animal testing for pharmacology and toxicology testing. Next is these studies are necessary for filing investigational new drug application. When will you go to file INDA or NDA? You need to represent the data of non-clinical or preclinical studies. You have to represent or show the regulatory authorities the data, because by this data they conclude if there is any toxicology of the drug. Next is need for clinical studies. What is the need of clinical studies? Same topic, to determine the lethal dose, toxic dose. Lethal dose is LD50, ED is effective dose 50. It means the lethal dose 50%, half percent in the population, and effective dose is half percent. It means 50% effective dose in half of the population. To determine the pharmacological action of the targeted drug, it means when we will after doing and testing on animals, we can conclude the results of pharmacological action of the targeted drug. It is necessary to submit the animal testing report during the IND application. IND application is investigational new drug application. So, when we will go to submit the application for approval of any new drug, we need to represent the animal testing report, which is known as non-clinical reports. Next is to determine the pharmacokinetic properties of a drug. Pharmacokinetic means what the drug does to the body. Action of drug on the body. Next, to determine the route of administration. It means after testing on animals, we can conclude which route is best for this medicine or that medicine. It means the route is maybe oral, sublingual, chewable, intravenous, intramuscular, etc. This can be concluded only after doing non-clinical studies, and we will use this data in clinical trials. Next is non-clinical drug development process. So, in non-clinical drug development process, there are mainly three processes. Number one, pharmacological profile. In pharmacological profile, we check the pharmacokinetic and pharmacodynamic. These two comes in pharmacological profile, and we conclude these results after doing non-clinical drug development processes, non-clinical studies or preclinical studies. Next is experiment on rodents. Rodents means mice. Then experiments on larger animals. After rodents, we do testing on big animals like monkeys, etc. Next is pharmacology of non-clinical studies. So, the pharmacology includes pharmacokinetics and pharmacodynamics. Pharmacokinetics means consists of three factors: primary, secondary and safety. In primary, we check the effect of new drug or the compound on the targeted organ. It means the compound, and we have to check its effect on the targeted organ. In second pharmacokinetic, we check the effect of compound but not on the targeted organ but the other organs instead of the main targeted organ. In primary, we check the effect of compound on the targeted organ, but in secondary, we check the effect of compound on other organs instead of the targeted organ. And the safety of use of drug in humans. Next is pharmacodynamics. Pharmacodynamics means action of, it means what the body does to the drug, means A for absorption, D for distribution, M for metabolism and E for excretion. When we take any drug, then first is absorption, then distribution, then metabolism, and last is excretion. Next is toxicology. There are various side effects and toxicity of any drug which are studied under toxicology. Next is toxicology. What is toxicology? It helps in determining the toxicity of compound. Any new compound on which we are doing non-clinical studies, we need to check the toxicity of compound. So, the toxicity check on targeted organ, poor pharmacokinetic properties, means action of drug on our body, and hepatotoxicity means toxicity of compound on liver. Hepato means liver. Next is toxicology studies. So, how do we do toxicology studies? The first is single dose and dose range finding studies. When we do single dose and dose range finding studies, we check to identify target organ of toxicity, reversibility of the toxic effect, dose for future reference, maximum tolerated dose, non-observed adverse effect level. It means in single dose and dose range finding studies, we identify what is the target organ of the toxicity, which is the main target organ of toxicity of compound, and reversibility of the toxic effect, if the toxic effect is reversible or not, and doses for future reference, how much dose we can tolerate so that the compound does not show toxicity, non-observed adverse effect level. Next is genotoxicity. As the word implies, geno, it means is related to gene mutation or DNA or RNA. It means if the toxicity leads to any gene mutation or chromosomal damage is known as genotoxicity. Next is toxicology studies. Same topic, carcinogenicity, to detect if the compound causes cancer, if the new compound its toxicity leads to any cancer inside body. Next, reproductive and development studies. It means to study the effect of toxicity on fertility, fetus and lactation. If we do a study of toxicology effect of any compound during the reproductive and development stage, and uh, study the effects on fertility, fertility timing, fetus and during lactation. During lactation time, the toxicity can reach to the baby. Next is drug metabolism. What is metabolism? Metabolism is also known as chemical alteration of drug in the body. It means when we take any drug, then inside body there is a alteration of chemical compounds known as the drug metabolism. So, during drug metabolism there are many changes happens inside the body. Number one is active drug to inactive or less active metabolite. Sometimes we take any drug and it was active, and then after metabolism it became inactive or less active metabolite. For example, phenobarbital is more active than para-hydroxyphenobarbital. Next is active to a more active metabolite. It means codeine, example codeine to morphine. It means codeine was a active metabolite, active compound, but when after metabolism the new compound formed morphine is more active than codeine, obviously. Next is inactive to more toxic metabolite. Maybe if there was a compound which was inactive, then after metabolism inside body it can become more toxic metabolite. Next is inactive to an active metabolism. For example, phenacetin to paracetamol. After drug metabolism, the inactive form of phenacetin converts to active metabolism of paracetamol. Next is active drug to an active metabolite which is having different pharmacological activity. It means drug is same or compound is same, but their pharmacological activity is different. It means, for example, iproniazid to isoniazid. It means iproniazid has a different pharmacological activity, but isoniazid has a different pharmacological activity. Iproniazid, earlier known as antidepressant drug, but also used as anti-tubercular, but this is mainly used as anti-tubercular drug. So, the active drug to an active metabolite which is having different pharmacological active. This was also active, this was also active, but now they have different pharmacological actions. Now, phases of drug metabolism. There are two phases of drug metabolism. Number one is non-synthetic or functionalization phase 1. In this phase, metabolite may be active or inactive. Phase 1 reactions are oxidation, reduction, hydrolysis, cyclization and decyclization. Phase second, known as synthetic or conjugation phase. In this phase, the metabolite is generally inactive. The main important change between these two phases is, in phase 1 the metabolite may be active or inactive, but in phase second the metabolite is generally inactive. In phase second reactions, the reactions are glucuronide conjugation, acetylation, methylation, sulfate conjugation, glycine, glutathione conjugation and ribonucleotide synthesis. Next is advantages of drug metabolism. Advantages is, it is necessary for the termination of drug action, reduce lipophilicity and increases renal excretion. After drug metabolism only there is a renal excretion of the drug. If drug doesn't metabolize inside human body, then its renal excretion is impossible. So drug metabolism increases renal excretion. Inhibition of drug metabolism. The factors which inhibit drug metabolism inside body is, one drug inhibits the metabolism of the other drug. This is the term is also known as antagonism, in which one drug inhibits the metabolism or not antagonism, one drug inhibits the metabolism of other drug. Next is prolongation of drug effects. If the drug effect is prolonged, so there is inhibition of drug metabolism. Third one, increasing the circulating level show the metabolism of drug. Next is, thank you.